It looks like we're starting back after the break. Before I start introducing Dr. Shern, I also want to thank, just like Dr. Nguyen did, the Children's Cancer Foundation and the Kaler Foundation for hosting this symposium. It's so wonderful to see everyone, and I think the presentations have been really wonderful. I also want to thank our speaker earlier today, Alia, who really gave a wonderful presentation. So it's a great honor to introduce our keynote speaker, Dr. Jack Shern. Jack is a Lasker Clinical Research Scholar at the Pediatric Oncology branch of the National Cancer Institute. He obtained his MD from the Medical College of Georgia, and it is a residency in Chicago University. And then POB was really fortunate to recruit Dr. Shern to come as a fellow for his pediatric hematology oncology training. Jack trained for his research in Javed Khan's lab, and POB was then very, very lucky to keep him on as a Lasker Clinical Research Scholar. Jack has really built a phenomenal program in the POB. He's one of the most collaborative people I know. He is using cutting-edge genomic and molecular profiling techniques to understand solid tumors, in particular sarcomas, and there in particular rhabdomyosarcoma and NF1-driven tumors. He's dissecting the heterogeneity of the tumors, the clonal evolution of these tumors, and uses these techniques to develop better precision therapies. The first speaker today, Tara Santum, did a really nice job highlighting some of Jack's seminal work in rhabdomyosarcoma. I'm very happy that he's collaborating with my team on very difficult tumors to treat, neurofibromatosis type 1 related tumors. But beyond this, Jack is very actively engaged in the Children's Oncology group and at the NCI level in something else Tara mentioned earlier today, the Childhood Cancer Data Initiative, and there in particular the Molecular Characterization Initiative, and also in an effort that we have in the Pediatric Oncology branch in working on our My Pediatric and Adult Rare Tumor Network. Jack is a wonderful doctor, a great scientist, collaborated, and really a gifted speaker. When I recently received some communications training, I mentioned Jack's name, and they said, yes, he had the training, but he doesn't need it. And I think you will see this in his talk today, Targeted Treatment Strategies Based on Understanding of the Molecular Mechanism of Tumors. Thanks for welcoming Jack.
Okay, well, that makes it sound much more interesting than it is, but I really appreciate that, Brigitte. And I'm just thrilled to be here today, and I want to thank the organizers of the event for having me. This is actually the first keynote that I've given, and what they've told me from the keynote is you get some extra time to give your personal philosophy, and that's what I'm going to do today. And I see my old friends rolling their eyes right now because they know when I get a microphone, sometimes I don't stop. So I hope today to tell you about the idea of mechanism. And I think this morning you've heard from multiple speakers now highlighting how they look at mechanism and how important mechanism is to what we do as we look for new therapies to treat these patients. And so from where I sit in my laboratory, in my clinical sphere, I see two strategies right now for how we can make progress for treatment of these patients, most of whom have very rare tumors and who have very few options available in the clinic. The first is to improve the accuracy of how we diagnose these, how we classify disease, how we stage disease, and how we detect disease. And this is the basics of the clinic to me. If you can't make the right diagnosis for a patient, it's very hard to give them a targeted therapy. This foundation, CCF, has been very generous in helping us work on a cell-free DNA assay that we've built for patients with neurofibromatosis. And I think you heard Taylor Sunby in my group give that talk a couple years ago. Today, though, I'm going to tell you about this second point, which is really why many of us go into this field, and that's to develop new specific therapies. We want to treat these patients better. We want to fix things. We want to come up with new therapeutic strategies. And for this, I highlight those words that were given to me about what I was supposed to talk about today, which is mechanism, mechanism, mechanism. And after hearing the talks this morning, now I know why they wanted me to highlight mechanism, because there's just beautiful mechanism in the laboratories that you've heard from today. Lee Hellman taught me, as a very early mentor in my career, that mechanism is the key. If you don't have mechanism, Lee would never even do a mouse experiment. He would not take it forward into the mouse until you told him exactly how that drug worked, how the molecular mechanism worked, or how he expected that experiment to come out. And I took that and said, that sounds like that's pretty important. Lee usually has a pretty good idea. The one time in my career that I saw this change was when we were doing CD19 CAR T-cell trials, when they were first coming out. And Crystal Makel, who was another great mentor of mine, she said, Jack, I don't care about mechanism. Just give me what's on the cell surface and I will fix it. You know, if you follow Crystal's work in the 10 years since, it's all very detailed mechanism, because you cannot leave it behind. You cannot separate it from what we do as we develop new therapies. So mechanism, mechanism, mechanism. I'll give you an analogy. Why do we need to understand this mechanism? This is my ride right here, a 1999 Lexus RX 300. It currently has 323,000 miles on it. And I am very good friends with a mechanic named Mike in Kensington, who spends a lot of time with my vehicle. I like Mike for a couple reasons. Number one, he's a philosopher. He likes to tell me about things that he finds in cars and how it affects his life. So we spend a lot of time talking about this. I also like Mike because he can fix things. I'm pretty sure, given this vehicle and his ability to keep it running, he can fix anything. And the reason why he can fix anything is because he understands every detailed mechanism about this car. He understands every part, how it moves, how it interacts with the other parts, how he can fix it, get to it, take it out, and put in a new part, and it will work the same way. And so, you know, I think this is how we should think about things, is if we don't have that detailed understanding of what we study, we will never be able to fix things. And so that's why I'm just so impressed with the talks this morning, because everyone is digging deep into mechanism. But mechanism can be overwhelming. So you heard this morning, you know, there is just a very detailed story behind every one of these mechanisms. And this, to me, is why I think these seminal papers that have been written over the years by these two gentlemen, Hanahan and Weinberg, which they call the hallmarks of cancer, they really set the framework for how we should think about mechanism. Because as you see here, in 2000, they had already made the point that, you know, really this could continue and continue and continue by adding layers of complexity to the scientific literature. And really, even in 2000, that was sort of beyond the complexity and the ability to understand things. So what we really needed to do was step back and build frameworks for how we're going to look at cancer and tumors. So rather what they saw is, from this complexity, you would start to have understandable small numbers of basic principles. And again, this morning's talks sort of highlight a couple of these. So they started out with 6 underlying principles. You heard from patients this HRAS oncogene. Well, that's that green bar right there, that self-sufficiency and growth signal. It's a common feature of multiple cancers. You heard the Ezrin story, tissue invasion and metastasis. So these are common programs that every tumor will use, and that's how I sort of started to structure how I think about the complexity of cancer, which is really hard to understand if you look at all the details. The other point that they made in early 2000 is that this is a multi-step process, and that it doesn't have to follow the same program every time. When I look at this graph and this picture that they drew in this original paper, you can see that there's things that always happen early. So the green arrow there, that's that HRAS oncogene. It typically happens very early on in these timelines as these patients go on. But you can see that the sequence of events that happen that lead to the eventual tumor can be different. And this reminds me very much of if you've seen how kids today learn computer programming, this is how they teach it. They show different cassettes, and the kids figure out different ways to put those things together. So the cancer cell, to me, is sort of following a program and figuring out new ways to put parts together to get the end result, which is proliferation and growth. So the other point about this is that every patient could then follow a unique path. And that's a little daunting. Every patient's going to be a little bit different. So if you want to have a targeted therapy, that's a little bit daunting. But I think the underlying programs follow a path. And that's what we have to understand when we think about our cancers. This is the next paper that these guys wrote about 10 years later. They called it the next generation. And here they've added 4 new categories. The first one there that you see is deregulating cellular metabolism, genome instability and mutation. And then look on the right-hand side now. 2 things that Rosa just talked about: avoiding immune destruction, tumor-promoting inflammation. So these now become new hallmarks. We're up to 10 hallmarks of cancer. If you understand these 10, you understand all of cancer. I think the point that they really highlighted here in 2011 when they were talking about this is that we can no longer take a reductionist view of tumor and tumor biology. And for me, I'm a very simple thinker. I take the reductionist view every time. I'd much rather study the cancer cell and understand specifically about the cancer cell. But people like Rosa in our group are teaching me very quickly that you really have to understand the immune system that's within the tumor and the fibroblast cell that's in this tumor, the endothelial cell that's in the tumor, and all of those are potential therapeutic options, and they're probably contributing to the growth of this tumor. So it's a major leap forward with this next generation paper. Okay, this one just came out in 2022. They call this New Dimensions. And you see now that Weinberg dropped off of this paper, and now it's just Hanahan. And 4 new features or hallmarks of cancer have been added: unlocking phenotypic plasticity, senescent cells, non-mutational epigenetic reprogramming, which we think a lot about in my group, and polymorphic microbiomes. I see people like Nico Josa in the audience getting excited thinking about things like that. So, I think while this continues to grow, this circle continues to grow of hallmarks of cancer, we're up to 14 here. We started at 6. I don't know that it's going to continue forever. I think we'll continue to sort of put things into individual bins, but I don't know that. And the deeper we dig into mechanism of individual tumors, the more potential we have to add new features that we can understand. So the other thing that I wanted to point out is that I'm a huge believer in technology, because most of these sort of new discoveries are driven by technology. And I learned this very quickly in my laboratory experience, especially in Javed Khan's group. I sort of came of age when next generation sequencing was coming online. 2010, 2011, we were able to sequence things in deeper and more significant ways. And you can see this machine here right about 2005, which was a 454 GS20. We had one of these in the laboratory, and it was really exciting. You could sequence a whole gene with this from start to finish. And it was slow, and it was clunky. It was always broken, but we could sequence a whole gene. And that was pretty cool. But at the same time, you had these new sequencers that could come out, and they could sequence the whole exome. And Javed was so far ahead of things, he even had things that could sequence the whole genome. And I was just sort of said, wow, I need to be part of this, because technology and being able to see the whole genome is a lot better than seeing one gene. If you pick one gene and go after it, you better be certain that that's an important gene. Otherwise, you're going to spend a lot of time working on that and not see the big picture, which is what I wanted to see. So technology is important. And you've heard this a couple of times. So I'm talking about 10 years ago when I was doing this in a research laboratory. Now these things are in the clinic. And that's amazing to me, is something like this, a national effort that we call the Molecular Characterization Initiative. This is a partnership between the Children's Oncology Group and the National Cancer Institute. And right now, any patient with a brain tumor who is seen at a COG institution can enroll and get their tumor profiled. And it's not one gene profiling, it's the whole exome. It's a methylation array, and it's looking for any of the fusions that we know occur in these tumors. And as you've heard, this is sort of coming online for the rest of the cohorts in the country. And these are clinical results. So these results go back to the patient. You don't have to go to a commercial operator. You don't have to wait for insurance approval. You enroll the patient on COG's Project Every Child. These results will come back to you in 21 days. And I'm excited to have this open up in the soft tissue sarcoma space, which is what I work on mostly. This will happen within this month. So at the end of this month, any soft tissue sarcoma patient, please enroll them on Project Every Child, and you'll get these sequencing results. Rare tumors will follow behind that. So I think this is sort of a massive opportunity for the entire community, and something that I've been a big proponent of. The other thing you heard from Rosa presented some beautiful single-cell sequencing data, but this, I think, is sort of another tool, another technology that's rapidly changing how we do experiments and how we think about things. So this is another one that's sort of exploded over time. And I started doing these assays when things were called DropSeq, about 2015. And at that time, you had to build your own single-cell sequencer and the microfluidic that went with it. And I'm always amazed by this original microfluidic because it shows sort of the power of this. So what you see down at the bottom of that little movie is a droplet of oil. It's like 1 nanoliter in size of droplet of oil. And what you see in the black, that big circle, is a barcoded bead. And what you can potentially see, if you look close enough, is down from the bottom, a single cell comes. And that single cell is captured with a barcoded bead in that nanoliter droplet of oil. So that's a technology, huh? That's pretty cool. And now we can sequence that individual droplet of oil along with the rest of droplets of oil that you can make from a suspension of cells. And so this is powerful. This is the ability now to look at every single cell within our experiments. And what you've heard is the data interpretation gets much more challenging. And also your experimental design, you have to be much more thoughtful about. But the way I feel about this is the way I felt about whole exome sequencing in 2010. It was a research operation to do whole exome sequencing back then. It took weeks to get the data back. It took even more weeks to figure out what the data meant. That's where we are with single cell sequencing right now. 10 years from now, we will routinely use this, and hopefully even on clinical samples, because I think it's that powerful. So what does this allow us to do? This is the analogy that I've seen before. You know, when we do bulk sequencing, we look at the entire tumor. That includes the immune cells, the fibroblasts, the stromal cells that are all within that tumor. And we grind it up and we look at the big mishmash of it. And we try and deconvolute that in some cases. But you can imagine that's pretty hard to do. What single cell sequencing allows us to do is then look at individual populations of cells and study them in detail. And again, giving us resolution that we have not had before. So that's sort of my philosophy part of the talk, and now I'm going to sort of tell you where we are working on a particular mechanism in this particular disease type that we have been working very hard on. And Brigitte mentioned this patient population, those patients that have this disease called neurofibromatosis type 1. This is a common disease. 1 in 3,000, 1 in 3,500 patients or people in the population have this. And this is driven by a loss in the germline, a germline mutation in a gene called NF1. And this, like the HRAS mutations you heard about this morning, cause a RAS pathway activation. So there's the green bar of the hallmarks of cancer driving this particular tumor. These patients have a really unique phenotype. They have some skin findings, cutaneous neurofibromas, which are benign tumors on their skin freckling. But what we've really been interested in as oncologists is this feature, tumor predisposition. Because what we know is if you have a germline mutation in NF1, you have a higher risk of getting nerve tumors, among other tumors, including leukemias and brain tumors. But what we've really focused on are these nerve tumors, the benign plexiform neurofibromas. We call them benign, but I'm not sure they are when you see some of these because they're large, bulky tumors in these patients. You can see in this patient here a large benign tumor known as a plexiform neurofibroma. And clinically, you have to watch these things because they can transform over time into a terrible sarcoma called malignant peripheral nerve sheath tumor, or MPNST. So the big problem for this patient population is there's really no effective medical therapies for the majority of these. Dr. Wiedemann and her group have been sort of seminal in finding MEK inhibitor, selumetinib, and getting that FDA approved for the treatment of plexiform neurofibromas. But otherwise, we really don't have great options for these patients. And the reason why I got interested in this disease is because the patients teach us some really fundamental characteristics, some really mechanistic details of how this disease progresses. And what you can see here is an MRI following a patient over time. And in sort of the first arrow there, you see the benign plexiform neurofibroma. Again, this is a bulky tumor, but it's really just going to sit there. It's not going to become a metastatic problem. These transition, if you watch them over time, into things called atypical neurofibromas. And then you can see sort of some focal development of the tumor by that third panel there, the arrow pointing to an atypical neurofibroma. We think these are benign, but starting to transform lesions. And if you follow those even further, within those, you will develop an MPNST. And somewhere about 15 to 20% of patients that have these benign plexiform neurofibromas over the course of their life will get this disease. And again, these teach us really important features about the mechanism, which is the importance of tumor cellular heterogeneity, as well as the mechanism of genetic tumor progression. And I'm going to talk about the second point here, which is the mechanism of genetic tumor progression. And the reason why I got interested in it to start my laboratory. So the first point is, these are really heterogeneous tumors. And I think Lindy's going to talk this afternoon at 4, so I'm going to set her up and say, we have to understand the tumor microenvironment better in these tumors. These things are composed of maybe 40% of immune cells, macrophages, T cells, dendritic cells. The tumor cells are a Schwann cell that's been transformed. And then there's a huge fibroblastic population that's making things like collagen and extracellular matrix. And each of these probably contributes at some point along this progression into driver of the patient's tumor's growth, as well as giving us new potential therapeutic targets, and also explaining why the tumors may even progress on therapy. So this is what's known about the tumor microenvironment of plexiform neurofibromas as of 2017. And Lindy's going to tell us more this afternoon about even more features that she's finding. And, I think, again, this patient population really teaches us this because these are huge tumors with lots of different cells. And understanding those interactions is sort of the future of this. But what really caught my interest as a tumor geneticist is this progression here and how well understood and well defined it was in many of the patients. And here you can see sort of the understanding right now of how these tumors progress. I told you these patients have one loss of one allele of NF1 in their germline. They still have one good copy, but we think this causes a RAS pathway activation. And some of these original works were done in 1990 by Francis Collins, who you've probably seen on TV. As I told you, these things can convert into plexiform neurofibromas, which is this benign second step. And the only genetic change that happens in that is you lose the second allele of NF1. So now you've lost both copies of NF1. Dr. Wiedemann and others have described this intermediate group, which is called an atypical neurofibroma. It's actually called multiple things by multiple people. ANUBs, is one term. I can't even come up with all the words that go under ANUB, but so that's why I call it atypical neurofibroma. And they're somewhere in the middle of this malignant transformation. And one of the only genetic findings that you can find if you look at these tumors deep enough is they'll lose one allele of CDKN2A. So another simple jump allows that tumor to start to progress. And then finally, if you start to look at the MPNSTs, it's sort of wild rearrangement. It's like the Wild West. The genome has become rapidly rearranged. There's lots of aneuploidy, gains and losses of chromosomes, but there's a recurrent one. Alteration of two genes, EED or SUS12, and these things are found in about 80% of all MPNSTs and maybe higher in the NF1 population. And these were first found in 2014, a group at Hopkins and a group at Memorial Sloan Kettering. And since that time, we've seen this recurrently in a vast majority of the MPNSTs that we've looked at. So we sort of said, well, that must be an important event. This is our progression model. We think that a Schwann cell will lose both copies of NF1. That'll form your plexiform neurofibroma. You lose that one copy of CDKN2A. Now the cell starts to cycle a little bit. You get that atypical neurofibroma. And then at some point, you lose either P53 or SUS12 or EED. You get a mutation in those genes, and the MPNST starts to form, starts to grow. So that's our current working model for the genetics of this. So what we really said was, well, this PRC2 mutation. So what is PRC2? SUS12 and EED are part of this complex of proteins that's a transcriptional repressive complex. We call it PRC2, polycomb repressive complex 2. And the whole job of this complex in the normal cell is to catalytically place this thing called H3K27 trimethylation. So we're getting pretty mechanistic here, but this is the job of this. And when this particular H3K27 trimethylation mark is placed onto chromatin, it closes the chromatin. So these are genes that are supposed to be repressed. So what does that mean when you lose SUS12 or EED? You can no longer place that H3K27 trimethylation. Now the chromatin opens and particular genes will turn on. And we said, well, that must be some sort of oncogenic program that the cell is trying to get to start to grow. And so that's what we really tried to study. You can see this within the patient's tumors. You can stain the patient's tumors for H3K27 trimethylation, and it's a very nice marker of malignant peripheral nerve sheath tumor. So what is known right now? What are the consequences of this? Some downstream assays have shown three things. Number one, you lose H3K27 trimethylation. I just told you that. H3K27 acetylation gain, so those are genes coming on, and H3K36 ME2 gain, same thing, genes coming on. Oncogenic signaling is amplified, so that NF1 mutation now has feedback which amplifies that. So that was shown nicely by Karen Tchaikovsky in Boston. And then there's some really cool work coming out of Children's Hospital of Philadelphia talking about the immunologic consequences of this. So if you lose PRC2, it seems like there's downregulation of MHC1 and decreased interferon signaling. So the cancer cell is now able to hide from the immune system. So this one mutation in SUS12 can do all of this. It seemed improbable. We wanted to study this in the laboratory a little bit. We have cell lines where we have MPNST cell lines that have lost SUS12. And you can see very nicely that they don't have H3K27 trimethylation. We built a doxycycline inducible system where you could put the SUS12 that's damaged back into that cell. And the idea was, okay, well, this is a simple experiment. We'll see what changes when you put the SUS12 back together, what genes come on and come off. So this is how I envision this. You lose SUS12, there'll be a certain set of genes that are on because you're not able to place that chromatin mark. In our doxycycline inducible system, we'll try and push that backwards. We'll try and put the complex back together, and we will repress the genes that the MPNST is trying to turn on. So this is RNA-seq from that experiment there. And what you can see is lots of genes change when you add the doxycycline, when you put the complex back together. In fact, there's about 1,600 genes that go down. These are probably direct targets of the polycomb repressive complex. But sort of paradoxically, about 1,300 genes go up. So I just put together a repressive complex and a bunch of genes went up. That's a little bit strange, but interesting, right? So what are these genes? If you do pathway analysis, which you've seen some of this before in the previous talks, you can get some hints to what type of genes are coming on and going off. And what we saw over and over again in every cell line we looked at was if you do this experiment, you get a lot of embryonic stem cell type genes that were being specific targets of the SUS12 polycomb repressive complex. A lot of very early genes, and in particular, it doesn't show up well here in red, but lots of transcription factors. This was sort of odd to us, things that were sort of stem cell-like transcription factors. Hox genes, Fox genes, things that were very developmentally regulated, seem to be regulated by this loss of polycomb in these tumors. And so the other sort of thing you're able to do with this is do chip sequencing. So you can actually find exactly where the H3K27 trimethylation mark is placed in this system. So here you can see minus docs. They really don't have any H3K27 trimethylation. If you add the docs, here's all the sites in the genome where this complex is putting its mark. And again, we saw over and over again that there's this group of key transcription factors, cell fate determining transcription factors that were really specifically targeted by this. And one in particular sort of jumped out multiple times in multiple cell lines, which is FOXC1. And here you can see the ChIP-seq tracks where you place that H3K27 trimethylation mark. The activating H3K27 acetylation mark goes down, and now the gene is repressed. So this seems like a very specific target of that polycomb repressive complex loss. And we had now a panel of these. We had about 43 transcription factors that looked very specific to this event. And we decided, well, let's just take all our cell lines and knock these out one by one. So this is a siRNA experiment where we're knocking all these transcription factors out one by one. And you can see in red highlighted the ones that are specifically regulated by that polycomb repressive complex. I think what you can see here in blue is that a lot of these, if you knock them down in the MPNS T cell, will decrease the cell growth. This is kind of what we were hoping for. This is the kind of target we're looking for. And here's some specific ones. Again, we were working very hard on this one called FOXC1. We had two different SIs. One knocked out the gene very well, and one knocked it out about half. And you can see in red there, if you knock it completely out, that's the curve you get for FOXC1. And if you knock it out about half, it slows it down a little bit. The same thing for this other gene we found as a specific PRC2 target, HOXB8. If you knocked it completely out, you sort of lost the cell growth. And we were able to go on then and ChIP-seq FOXC1 itself and sort of find where this was in the genome. And interestingly, FOXC1 went along with active genes. So anywhere where there was sort of very active gene expression, very open chromatin, FOXC1 was integrating and helping drive the expression of that particular gene. Here's TGF beta 1. So this, we think is sort of a circuit. A transcription factor gets deregulated by the loss of polycomb and then drives some downstream targets and helps to drive the oncogenic transcription of that cell. So I told you single cell sequencing is becoming more and more powerful. We've done a lot of patient tumors that have MPNST or plexiform neurofibromas and looked at their single cell profiling. And this is a particular patient here where we had lots of cells to look at. In purple there you see all, and I should say this is a UMAP plot, and every dot on this plot represents the expression profile of a single cell. So that's how you sort of look at this. And the power of this, you can see, now I can start to cluster out the cells from different populations. And you saw this in Rosa's talk too. It's actually pretty easy to tell apart an immune cell from a tumor cell. But you can also see that within the tumor cell, there's a proliferating population, and there's probably even other subclones within that malignant peripheral nerve sheath tumor. You can quantify this. We had 56,000 cells in this particular case, and you can see that a large component of this are T cells, a large component of them are myeloid, a large component are stromal. Again, coming back to that idea that we really have to understand how these things all interact. But if you just look at the tumor cells, and this is maybe the first time that you can specifically look at the tumor cell without collecting the information from all the immune cells and fibroblasts, you again saw this embryonic stem cell feature. And so this, again, I think sort of matched up with what we were already knowing. And if you look at the transcription factors that we pulled out of that in vitro experiment up front, you could see that these were very highly expressed within the malignant peripheral nerve sheath tumor. And this is quite reassuring because you're finding the same things in a patient sample that you saw in the in vitro setting. So we started to ask ourselves about this idea, and we see this over and over again in pediatric tumors, is this idea of de-differentiation. I keep telling you that it sort of gained this embryonic stem cell-like feature when it lost the polycomb repressive complex. So is this cell sort of a Schwann cell that's become a Schwann cell and now lost something that makes it a little bit more like a stem cell? That would explain why these things don't respond to chemotherapy. It would explain why they're so aggressive, why they like to spread and metastasize, like an early embryonic stem cell should. And if you look at the Schwann cell lineage, they come from the neural crest, and then they go through a precursor stage, an immature Schwann cell stage, before finally becoming that mature Schwann cell. And so we were sort of interested, does this polycomb repressive complex push it back towards these things? And again, leveraging single-cell sequencing, we were able to take publicly available single-cell sequencing from the normal trajectory of how a Schwann cell develops. And this is the map that we built. It matches up very well by gene expression profiling to what's known about how these Schwann cells develop in the normal case. And so this, I sort of envision as the map that we're trying to put our MPNST cell onto. And this is exactly what we did. And sure enough, when you start to look at the gene expression profile that we saw in the malignant peripheral nerve sheath tumor, and the expression profile that you can see in normal Schwann cell development, what you see is this group here, mesenchymal, a mesenchymal step in that migration from the neural crest stem cell. And if you look at the transcription factors that this mesenchymal cell expresses, FOXY1, PRRX2, PRRX1, TWIST1, FLE1, 6-2, and even the step right after that, that migratory 1, HOXB8, HOXA7, these are the same transcription factors we were pulling out of that polycomb repressive complex, specifically regulated transcription factors. So we think that what this cell has done by losing polycomb is really go back towards this mesenchymal feature. And it's doing this by releasing transcription factors that are known to regulate those steps in development. So this is a broken developmental program. If you looked at the hallmarks from 2022, one of the new topics is broken developmental pipelines. So this is another hallmark of cancer. And in malignant peripheral nerve sheath tumor, I think this is a very core feature of these. Again, we could use our marker gene sets from the malignant cells and from the normal Schwann cells in our single cell data to prove this in multiple different ways, even going as far as knocking out FOXY1 from these and coming up with a gene expression profile and linking that to what we saw in the patient tumor sample. So, I think in summary, as I sort of finish up here, how I envision this is that there is a loss of this component of this repressive transcriptional complex, and this kicks off a bunch of downstream events. And one of those is a transcription factor, maybe it's FOXY1, maybe it's HOXB8, and those feed back on themselves, go to open areas of the chromatin, and continue to drive this program that's supposed to only be on when a cell is a mesenchymal stem cell. And again, I think that matches up with what we know about these tumors, how difficult they are to treat, and why they are so metastatic. And so that's sort of where we are on this. And what I'll say is I have completely ignored there's another side to this. And I told you up front that there's an immune component where if you look at polycomb deficient tumors, somehow they've downregulated antigen presentation and interferon response. And I think what we're sort of approaching the ability to do is understand how this one mutation in SUS12 can not only upregulate this mesenchymal neurocrest stem cell program through these specific transcription factors, but it might also make it very hard for the immune system to see. And understanding this mechanism is the key to figuring out how we're going to fix this. I don't have the answers to that right now. What I do know is if we could knock any of these transcription factors down, the cell will die. And I think if you are able to do that, you'll be different than anyone else who's ever tried to drug a transcription factor. It's very hard to do. But again, I think if we use technology and the coming technology, we're going to understand how to do this better in the clinic. And we're going to understand how to pharmaceutically do this. And I think that's sort of the next generation. How do we drug these things that are hard to drug? And once we have the mechanism, we understand that. And so I'm going to stop there and acknowledge a postdoc in my lab, Xiwan Zhang, who's done all this remarkable work on these transcription factors and this transcription factor circuit that underlies this MPNST. I'd like to thank all the members of my group that really help drive some of these projects along. Members of the Pediatric Oncology branch, as well as lots of collaborators across the country. And then, of course, the work that I do is absolutely not possible without the patients and the families. We learn so much from every patient's sample and every patient's clinical course that I just think it's such a critical team effort to work on these tumors, and I thank all the patients and families for their efforts. So I'll stop there and take any questions.
Before I give him the microphone, I just want to say that was an amazing talk, Jack, with just a beautiful introduction and an overview that I think hopefully everybody has both a new appreciation for context and also an appreciation for complexity. So that was really good. I want to ask you a quick question, though, before I give it to Dr. Rubens, because I'm holding the microphone, I get to do that. Okay, so you don't know how to do it. You've got this list of 6 transcription factors. Use your imagination. If you could come up with some ways of doing this or ideas that you'd want to see go forward, how would you do it?
Yeah, I think it's, like I said, an exciting time. There's, in my group in particular, we're working very hard on this technology called PROTACs. And that's an idea where you can find a small molecule that will bind a protein of interest and then drag it to some of the machinery in the cell that will degrade it. And some of these have been fairly powerful. We're working with a collaborator at the Broad who's actually developed one to target MYC. And they don't always work the way you think they should. And it's pretty hard sometimes to drug these because, as you know, they're kind of floppy and the protein structure isn't great. But I think it is going to be doable. And so we're going down the PROTAC route right now. I also have a fellow who's joining the lab who's got a lot of experience with genetic manipulation of these. So delivering targeted siRNAs or targeted ways to degrade the RNA would be another strategy. But I think these things are coming, and I think a lot of people are thinking about these now in different ways. So that's where
Cool.
I think the future is.
Thanks, Jack. Really, really great talk. It's exciting to see how you've used the technology to further understand the mechanisms in these diseases. I was wondering, so you see abnormalities in both SUS12 and EED in these cancers. Do you see them in both in one specific cancer, or is it either or? And do you think the mechanisms are redundant or similar, or how the abnormalities are different?
I think so far, when we've looked, they're mutually exclusive. Mm-hmm. But, you know, it's a struggle in a rare tumor to come up with enough tumors to look at. And, you know, with great collaborators at Hopkins, at WashU, we're able to start to put cohorts together. But if, I look at my rhabdomyosarcoma experience where we had 600 rhabdomyosarcomas, and we don't even think we've sort of explained all the mutual exclusivity of these. I just think we're going to need larger numbers. I don't doubt that the tumor can fool us and have both a EED mutation and a SUS12 mutation. Tumors do wild things in the genome.
So Jack, I really enjoyed your talk a lot. I think you definitely delivered. It's a beautiful, beautiful talk. I have a question as it relates to treatment. So do you think if you were smart enough to attack the PRC2 complex, would that be all you need, or do you need additional immune-targeted therapies? Do you really need a combinatorial approach?
Yeah, I mean, I think even if you go back to that 2000 Hallmarks paper, a combination approach just makes sense. When, I said, it reminds me of kids' computer programming, where you have different modules. I think the tumor's got a lot of different modules that it's using to meet the eventual goal for that cell, which is growth. And so, we're going to have to think about ways to target different modules at different times and in different combinations. And, these cells already have an NF1 mutation and no NF1, activated RAS signaling. Maybe we probably need to start there because we'll probably have to turn that off as well. So I think, again, having a mechanistic picture of the big picture is the only way to start to rationally put those things together. And that's, I think you're right. I think we'll need lots of combinations. The other thing, that I think in the clinic we're seeing over and over again is, use a different mechanism completely. And that's why I think immune therapy has been such an amazing thing, because now we have a whole different modality to attack. And the toxicities don't necessarily overlap with how we do things with chemotherapy or other drugs. So, we have different ways to mix and match, and that's where the future is there.
Great. Oh, Charles.
Thank you. Really nice talk. I actually had two questions regarding this sort of dedifferentiation and developmental plasticity. The first is that dedifferentiation into the mesenchymal stem cell, is it completely dependent in a cell autonomous way on EED or SUS12 mutation or PRC loss, or could there be paracrine effects that allow other tumor cells to dedifferentiate?
Yeah, I mean, I think that's pretty much unexplored right now, but it's probably true that, if you look at the growth factor composite of these tumors, these plexiforms have sat there for a long time and created a microenvironment. I think there could very well be paracrine signaling too. I think we've sort of focused on that PRC2 specific side of it to see if there's anything there. But
I mean, from the sequencing, do you ever see, from the single cell seq data, do you ever see cells that have that expression phenotype that don't have the mutation in those populations?
Yeah, I mean, there's hints of it. But the one thing that I'll say is I talked up single cell sequencing as if it's a cure-all. The one thing about that is the data is really sparse. And I think, it's hard to make inferences like you can see patterns, but when you start to dig down into individual questions like that, you have to be really thoughtful how you do that. So I think it's possible.
Thanks, and I guess my second question also probably may push beyond what your data can let you know, but I'm curious how your sense of how plastic and ongoing these transition states are. Once it dedifferentiates, how static is that versus, how actively is microenvironmental factors or other factors driving things to move back and forth in their differentiation?
Yeah, so it's actually pretty remarkable because we have several cases where we have a primary and a metastatic lesion. And you can see actually the tumor cell changes from the primary to the metastatic site. And a lot of that is downregulation of immune components. And so I think there's lots of mileage there for figuring out, okay, is the microenvironment of the metastatic site different than the primary site? Maybe we shouldn't be so focused on that, but maybe the metastatic site is really where the money is. And we do see that. We see differences between the primary MPNST cell and then when it goes to live in the lung as a metastasis.
Thank you so much.
Cool. Other questions? All right. Well, thank you again, Dr. Shern.